ఇప్పుడే స్టార్ట్ చేస్తాను. ఇప్పుడే స్టార్ట్ చేస్తాను. కొన్ని చెప్తూనే ఒక్కొక్క లైవ్ అంటే చాలా ఉండేది.
So started. Good afternoon, students. Myself, C. Raja Ramesh, Zoology Senior Faculty, Hydernagar, PVTG, Girls. So today we have discussed about that RH blood typing and as well as Erythroblastosis Foetalis. So previously we have discussed about that blood groups. In that blood groups, there is a 4 type of blood groups are identified by Carl Landsteiner in 1900. They are A, B, AB and O blood group. So in that blood groups, you have already discussed about that there is a positive blood group and negative blood group. So how you can define that positive and the negative blood groups? So apart from that of ABO blood groups, again the Carl Landsteiner and Alexander S. Weiner identify the another one type of antigen in the blood of rhesus monkey. So that antigen is known as the RH antigen. That monkey name, scientific name of monkey, it is Macaca mulatta. So right now here it is we can discuss about that RH type antigen. Here it is a RH typing. Carl Landsteiner and Alexander S. Weiner scientists are discovered the RH antigen in the blood of a monkey. In the blood of monkey, that scientific name of monkey, it is Macaca mulatta. Macaca mulatta. Right. So in the blood of monkey, there is a RH type antigen, it is the identified. So after that, this one RH antigen, it is also identified in the blood of the human being also. So after that, this one RH antigen, it is also identified in the blood of the human beings, especially that is, here it is the RBC. The RBC, it is includes that plasma membrane. On the plasma membrane, there is a RH antigen, it is identified. So later on in the human being, this one RH antigen, it is also known as the D antigen. So here it is the known as the D antigen. So those persons are have the D antigen, so they are known as the RH positive persons. So those persons are divide the RH antigen means D antigen, they are known as the RH negative persons. So RH positive means D antigen is the present and RH negative means D antigen, it is the absent. So generally throughout that worldwide, so generally in Indians, so there is a 80% of humans are the RH positive persons. So here it is the 80% humans are concerned to the RH positive. So here it is a general information according to this RH typing. So there is a RH blood group, it will be according to that Fisher and Reis scientist. According to the Fisher and Reis scientist, there is a antigens are identified. So according to Fisher and Reis hypothesis. So here it is the Fisher and Reis hypothesis. Fisher and Reis hypothesis. The Fisher and Reis scientists identify the sum of alleles on the chromosome 1. So here it is the chromosome 1. So in the chromosome 1, there is a sum of alleles are the identified that alleles are the concerned to the Rh antigen or the D antigen. So here it is a chromosome 1, it is included that 3 loci. So each and every loci it is included that alleles. So here it is the either dominant allele, either there is a either dominant allele or either there is a recessive allele. In the homologous chromosomes, there is a either dominant allele in the form of the C or in the form of the small c, either in the form of the capital D or the small d, and capital E and a small e. So here there is a 3 are dominant alleles and 3 are the recessive alleles are the present. So according to this one, 3 alleles, 3 pairs of alleles, so we can find out the different type of the genotypic composition. So what are the genotypic composition according to the rhesus in a rhesus monkey, according to the Fisher and Reis scientist. So here it is the C, D, E, R, small c, small d, small e. So this is the one composition, and second one it is there capital C, small d and capital E, and then after there is a small c and capital D and a small e. This is the one, and there is a here it is the small c, small d, small e, and as well as small c, small d and a small e. Then after capital C, capital D and a small e, and as well as capital C, small d and a small e. So here we find out the 8, that means 4 pairs of the alleles are you can find out. By this one Fisher and Reis hypothesis, so we can say that, so which group it is the includes that capital D, so that group it is identified as a Rh positive. So which group it is included that small d, that is a recessive in condition, we can say that it is the Rh negative. So according to the Fisher and Reis scientist, the Rh blood typing it is identified in the name of the CDE nomenclature. By this one CDE nomenclature, we find out that 3 types of alleles in the form of the dominant and as well as the recessive. So in the homologous chromosome 1, there is a capital C, small c, capital D, small d, capital E and small e's are the present at the particular of the loci. So these alleles are, they are forms the 4 pairs of the genetic composition. So this genetic composition it is includes that, it is explains that Rh positive and as well as the Rh negative. So in this genotype, so which is have the capital D, they are nothing but known as the Rh positive. So in this which is have the small d, they have that Rh negative. So here it is the known as the Fisher and Reis hypothesis, and according to that Weiner hypothesis, according to Weiner hypothesis, so there is a 8 alleles are the present. 8 alleles are the present. So in these 8 alleles, the R1, R2 and RO and RZ are Z, and then after here it is the R, small r colon and small r semicolons and small r y. So here in this one, so capital R it is indicates that Rh positive, and there is a homozygous recessive, either it is the small r or either small r colon either small r semicolons, either small r y. So all these are indicates that Rh negative blood group. So according to that Wiener hypothesis, there is a 8 alleles are the present, means 8 pairs of alleles are the present. In this one 8 pairs of alleles, 4 pairs are concerned to that capital R, and the another one 4 pairs are concerned to that small r. So capital R it is indicates that presence of Rh antigen, and the small r it is indicates that presence of it is the absence of the Rh antigen. Means absence of Rh antigen means it is nothing but known as the Rh negative blood group. So here the Fisher and Ray scientist and as well as the Wiener scientist explains that so Rh typing in the human beings. So here now we can discuss about that there is a erythroblastosis fetalis condition. So here it is the erythroblastosis fetalis. Erythroblastosis fetalis. Erythroblastosis means the formation of the immature blood cells in the fetus. So the formation of the excess number of the immature blood cells, immature RBC cells, it is nothing but known as the erythroblastosis fetalis. So this one erythroblastosis fetalis, it is that takes place according to that presence or absence of the Rh antigens. So here generally we can say that Rh positive person when marry the Rh negative person. So Rh positive person, for example here it is the male, and Rh negative person it is the female. So this marriage it will be takes place to the death of the second baby or the third baby or fourth baby. So whatever it may be. So first baby it is safe, but remaining of babies are the lead to the death. So here it is the Rh positive it is the men, that means male, and Rh negative it is the female. So here it is does not acceptable to the marriage of the people. Second one case. So here it is the when, so male it is the Rh positive and female it is the Rh positive, it will not any problem to the babies. So right now that means it is acceptable marriage. Second one, third one. So when men it is the Rh negative, male it is Rh negative, and female it is the Rh negative, female it is the Rh negative. So it will be compatibility it is takes place, there is no mismatching it is takes place. So that's why it is also acceptable marriage. So that means either it is a first child or the second child or third child, it is does not lead to the death, it will be safe. Right now here it is the fourth one. The male it will be, male is the Rh negative and female it is the Rh positive, female it is the Rh, female it is the Rh positive. So this is also acceptable to the marriage in between this one individuals, either first child or the second or the third, whatever it may be the children's or the fetus, it is not lead to the death. So which marriage it is does not acceptable means, so the male it will be Rh positive and the female it is the Rh negative. So here it is the first child it will be safe, but the second child it will be lead to the death. So how it is that takes place, we can discuss about this one clearly. Right now here the Rh negative male, so here it is the Rh negative female marry with the Rh positive male. Right now here it is the, already we can discuss about that according to the Fisher and Ray scientist. So what is that, here it is a capital D, it is indicates that Rh positive, and a small d it is indicates that Rh negative. Same thing you can write about this. Negative means D, small d, positive means here it is the capital D, capital D. For example, right now here the female organism, it is the leads to the production of the two gametes. The one gamete is includes that small d, and the second one gamete it is includes that small d, and the men it will be releases that two gametes, means it is the sperms. The one sperm it is includes that capital D, the another one sperm it is also included that capital D. It is allele for the Rh blood typing. Now these gametes are put into the phonate board. So here it is, how many gametes are there? 4 gametes are there. So in this one 4 gametes, just you can take the 4 boxes, 1, 2, 3. So here the left side you can take the male gametes, capital D and capital D, and female gametes are small d and small d. Right now, so the union in between the sperm and ova, capital D and small d, capital D and small d. So always already you can discuss about that Fisher and Reis scientist. So when the D it is the present, you can say that Rh positive. Small d it is known as the negative condition. Now here it is the individual or the child it is formed. This one child it is indicates that capital D. Capital D means positive for Rh antigen. Then after sperm and ova, here it is also capital D and small d. So capital D means it is indicates that positive right now. Then after the sperm and ova, here it is also capital D and small d. So here it is the positive it will be formed. And then after capital D and small d, capital D and small d. So here when the marriage it is takes place in between the negative female and positive male, all the children should be positive in condition. All the children should be positive in condition according to the Fisher and Reis scientist. Already Fisher scientist explained that the capital D it is indicates that Rh positive. The small d it is indicated that Rh negative. So here we are found that of the 4 children for this one couple, and the 4 children are expressed that positive in condition. So these positive children are lead to the hemolytic anemic disorder, or nothing but known as the erythroblastosis fetalis. So how it is that takes place? How it will be takes place? So in this one 4 children, the first one children it is the safe, but the remaining children are the lead to the death by the disease of hemolytic disorder of the newborn baby. So how it is that takes place? So how it is the takes place? Just you can see this one. So here it is the Rh negative female, and there is a Rh positive male. Rh positive male. So right now Rh negative female, Rh negative female. Okay, right now when here it is the indicates small d and small d, small d and small d, and here it is the capital D, capital D, for example. Okay, these are the gametes. Right, when the ova it is the fertilized by this one capital D sperm, it will be lead to the formation of embryo. So here it is the, for example, this is the embryo, and this is nothing but known as the uterus. For example, uterus. The uterus it is connected to the fetus through the placenta. Through the placenta. So here the Rh negative female when marry the Rh positive male. So in this one, the male it is produces the D, capital D sperms, and the female it is produces the small d. Here it is the small d, small d, small d. Okay, right. Small d ova. So when the small d ova, it is fertilizes with this one capital D sperm, it will be lead to the formation of the capital D and the small d embryo it is the formed. Capital D means it is nothing but known as the positive. So that means positive fetus it is the formed. That means in this one fetus there is a RH antigens are the present, but the mother it is the negative for antigens, negative for antigens. At the time of the supplying of the food through this one placenta, the mother it will be supply the food, the fetus through this one placenta. So at that time the baby it is the growthing it will be takes place. So after that growth of this one fetus, now at the time of the delivery, now this one fetus it will be safely released. At the time of the releasing, when the placenta it is ruptured, now the antigens of RH of fetus, they are mixed with the blood circulation of mother. Means here it is the positive condition it is the takes place in the mother's blood circulation. So at the time of the delivery of this one positive fetus, in the time when the placenta it is ruptured or hemorrhage it is takes place, by that of hemorrhage these RH antigens are mixed with the blood circulation of the mother. Okay, so right now slowly, so this D antigen means RH antigen, against of this one RH antigen the mother's immune system it will be sensitized. So the B cells of the mother, it will be produces the plasma cells and as well as the memory cells. So the plasma cells are the produces the RH antibodies. So what are the produced? RH antibodies are the produced. So these are nothing but known as the D antibodies. So here it is the what are the produced? D antibodies are the produced. So listen carefully. When female it is the negative for RH antigen or the D antigen, and here it is the male it is the positive for RH antigen. So how we can say that positive for RH antigen? Because that RH antigen it is present in the form of the capital D. These are the alleles for the presence of the RH antigen. Now here it is a small d means it is a negative for the RH antigen. When this one ova, it is the fertilizes with this one capital D sperm, and it will be lead to the formation of the zygote. That zygote it is the development as a embryo. Such embryo it is the capital D and the small d. The presence of the capital D, you can say that the fetus it is the RH antigen plus. That means positive fetus it will be developed inside of the uterus of the mother. Okay, right now. So through this one placenta, the mother it is a supply the food materials. Now the fetus it will be exhibited the growth. After the completion of the growth, after the completion of the gestation period, so this one positive fetus it will be delivered, or the parturition it will be takes place. At the time of the parturition of this one fetus, when the placenta it is ruptured, so these RH antigens are enter into the mother's blood circulation. Right now, so already what is the released, what is the delivered? The fetus it will be delivered. But at the time of the delivering of this baby, so mother it is the sensitized with this one RH antigens. That antigens are came from this one fetus. Okay, right now. So here it is that these RH antigens are, so here it is the antigens. When the antigens are the present in the body of the mother, now the mother's immune system is sensitized with this one RH antigens, and sensitization leads to the stimulation of the B cells. The B cells are the produce the plasma cells and memory cells. Now these plasma cells are produces the RH antibodies, or you can say that D antibodies. Okay, after the delivery of the first baby, there is a Rh antibodies are the released into the mother's blood circulation, or the D antibodies are the released into the mother's blood circulation. Okay, right now. After that 6 months, or the after that 6 months, again this one female organism, it is undergoes the pregnancy. That means it is a conceive that second pregnancy. So in this one, so right now again this one ova, it is a fertilizes this one sperm, fertilizes this one sperm. So here it is the mother, and this is the uterus, and this is the fetus. For example, this is the fetus. What type of fetus it is that develops inside of the uterus of mother? So small d and capital D. So here it is the capital D and the small d. Capital D means it is nothing but known as the positive. Okay, already according to the race and Fisher scientists, we can say that. So the D, it is indicates that Rh positive, and small d it is indicates that Rh negative. According to this one Fisher and race scientists, so the fetus it is formed capital D and small d. So capital D is the present now, so that's why it is nothing but known as the positive for Rh antigen. Now this fetus it is connected to the mother uterus. Mother uterus. Okay, at the second time pregnancy, in the mother uterus, the fetus, positive fetus, it is formed by this one condition. Okay, right now. So already what are the developed in the mother's blood circulation? There is a Rh antibodies. Okay, Rh antibodies are the developed, or you can say that D antibodies are the develops. So what are the develops? Rh antibodies are the D antibodies are the develops. So now these D antibodies are Rh antibodies through the blood circulation. So here these D antibodies are the small in nature. Small in nature. Okay. Now by the small nature, along with the food materials, along with the blood circulation, from the mother blood circulation, it will be enter into the fetus blood circulation through the placenta. Through the placenta. So right now the fetus it is exposes to the D antibodies or Rh antibodies. D antibodies or Rh antibodies. Now these D antibodies are react with the fetus antigens. So what are the reacted with the antigens of the fetus? The D antibodies are the reacted with the antigens of the fetus. So this reaction it is that leads to the lysis or the destruction, lysis or the destruction of the RBC of this one fetus. So here see this one clearly. You can see this one, you can draw the clear one diagram. Here it is. So here it is the fetus. Here it is the fetus. The fetus it is connected to the mother uterus. So this is the mother uterus, and this is the fetus. What type of a fetus it is developed in the mother uterus? There is a capital D and a small d. Capital D is the present now. The presence of the capital D, it is indicates that Rh antigen is the present. Rh antigen is the present, mean it is nothing but known as the positive fetus. It is the second pregnancy. So already after the delivery of the first baby, the mother it is the receives this one Rh antigens. Against of this one Rh antigens, the mother immune system it is that develops the Rh antibodies or the D antibodies. When the second pregnancy it is that takes place, so these D antibodies are enter into the fetus blood circulation. So here it is the enter into the fetus blood circulation. So because these D antibodies are the small in nature, the micro size in nature. So by this one small in nature, the placenta, it is allows the entry of the D antibodies into the fetus blood circulation. When these D antibodies are enter into the fetus blood circulation, these D antibodies are react with the D antigens or Rh antigens. So it will be leads to the lysis or destruction, lysis or the destruction of RBC of a fetus, RBC of the fetus. So here it is the excess destruction or the lysis of RBC of the fetus, it will be leads to the anemic condition. Anemic condition. Okay, anemic means why the anemic condition? Because of that destruction of RBC. That means destruction, it will be leads to that decreasing of RBC. When the RBC it is decreases, it is nothing but known as anemic condition. So that means the corpuscles, red blood cell corpuscles or the RBCs are the decreased in number. So that's why it is nothing but known as the anemic condition. Such anemic condition, it is nothing but known as the hemolytic anemia. Hemolytic anemia. And it is also causes that jaundice. It is also causes that jaundice. Okay, right now. So by this one hemolytic anemia, our body immune system, it will be try to overcome of this one less number of RBC. So in this one time, our bone marrow, okay, by this one disease, there is a number of RBCs are the decreased, but our body, it is a try to overcome of this one hemolytic anemia condition. So in this one condition, the bone marrow, liver and the spleen, they are try to production of RBC. Production of RBC. These RBCs are immature in condition. That immature RBCs are known as the erythroblast. The immature condition of RBCs are known as the erythroblast. So this one erythroblast condition, it will be takes place in the fetus. So that's why this event or this one phenomenon, it is known as the erythroblastosis fetalis. Okay, right now. So here it is the known as the erythroblastosis fetalis in condition. Okay, right now. So we can see again this one topic, erythroblastosis fetalis now, right now. So against of this one erythroblastosis fetalis, so here it is the erythroblastosis fetalis. Okay, this one erythroblastosis, why you can say it is erythroblastosis fetalis? Because of that excess of RBCs are the produced, that RBCs are immature in nature, that immature RBCs are known as the erythroblast. It is takes place in the fetus. So that's why its condition is nothing but known as the erythroblastosis fetalis. So how we can overcome of this one problem? How we can overcome of this one problem right now? So here it is the how you can overcome of this one problem. So at the time of first pregnancy, at the time of the first pregnancy, 28th week or 34th week, at the time of the first pregnancy, 28th week or the 34th week of first pregnancy. So we can given the IgG anti-D antibodies to the pregnant mother. Okay, such a time, so we can overcome the problem of the erythroblastosis fetalis. Okay, right now. So when you can see this one, when this type of marriages are that takes place, when this type of marriages are that takes place, the first baby it will be safe, but second baby it is suffered with the erythrolytic anemia, erythrolytic anemia or erythroblastosis fetalis, erythroblastosis fetalis. Second baby it is the suffered with this one erythroblastosis fetalis. So prevention of the second baby to disease at the time of the first baby, at the time of the first pregnancy, that means 28th week or 34th week of the first pregnancy. So what type of antibodies are given to the mother? There is a IgG anti-D antibodies are given to the mother. So when you did not given these IgG antibodies, D antibodies to the mother at this one time, so next chance it is also there. When the first baby or when the first baby is the delivered. Okay, so immediately delivering of the first baby, immediately you can given the anti-D antibodies, anti-D antibodies. Okay, right now. So here there is a two chances are there to prevention of the erythroblastosis fetalis in the second child. So what is the prevention methods? The erythroblastosis fetalis in the second child. So at the first pregnancy, the 28th week or the 34th week, so there is a IgG anti-D antibodies are given to the mother. Okay, so these are suppress the Rh antigen reactions. So that's why the second baby it will be safe. And otherwise, so after the delivery of the first baby, immediately you can given the anti-D antibodies to the mother. So it will be suppress the antigens in the Rh antigens in the second pregnancy. So that means second baby it will be safe. So here it is the importance of this one, erythroblastosis fetalis. Right now, here it is the in this one you can see this one according to the Fisher and race scientist. So how many alleles are identified? There is a 3 alleles are identified. Na? So one it is the D, that means C, D, E, or either small c, small d and a small e. Where these are present? These are present on the chromosome 1. So here it is the chromosome 1. This is the chromosome 1. So the chromosome 1, it is how the locations. There is a 3 locations are present. In these 3 locations, there is a capital C or either small c is present. Capital D are present or small d it is present. Capital E present or small e it is present. Same thing it is takes place. Capital C or the small c. Capital D or the small d and capital E or the small e is there. So according to this one 3 alleles, we can get the 4 pairs of genotypes. So what are the genotypes? So here it is the capital C, capital D, capital E. So here it is the capital C, capital D, capital E. Either small c, small d, small e. Small c, small d and a small e. Okay, then after. So capital C, small capital C, capital D, okay, capital C, small d, capital E. So here it is the capital C, small d and small e. Here it is this one, C, D, E. Okay, right now. Then after, here it is the small c, capital D, small e. Small c, capital D and small e. Then after, here it is the small c, small d, small e. Small c, small d, small e. All these small c, small d, small e. Small c, small d, small e. Then after, capital C, capital D, small e. Capital C, capital D, small e. Capital C, capital D and a small e. Then after, capital C, small d and a capital E. So here 1, 2, 3, 4 pairs of alleles are the present. In this one, so the D, this is a nothing but known as a positive. So here it is the nothing but known as all the small letters are there. So this is nothing but known as the negative condition. So where the capital D it is the present, it is indicates that RH positive. Where the recessive homozygous it is the present, it is indicates that RH. 1 minute. So where the small d it is present, where the small d it is present, it is indicates that RH negative right now. Then after, so here it is the Weiner scientist, it is also proposed that another one theory na? So that theory, according to that one theory, there is a 8 pairs of alleles are the present. So these, by this one also you can say that RH antigen and RH and RH antigen is the present or absent. So here it is the that one R1, R2, okay, RO and RZ. Second one R colon and semicolons and RY. Okay, now the presence of capital R, it is indicates that presence of the RH antigen. Okay, right now here it is a homozygous condition, either RR or either R1 or either semicolons, either RY. So these are indicates that absence of the RH antigen. RH antigen means you can say that here it is a, when the ova it is how this one capital R, ova it is how the capital R, so it is the RH positive. When the ova it is how this type of condition, you can say that it is the RH negative. So when the sperm it is how the RH R or RZ, you can say that it is the positive. When the ova it is how the R small y, you can say that it is the negative condition. So according to that Fisher and Reis scientist, and according to that Weiner scientist, you can say that RH positive and RH negative. So this is nothing but known as the RH typing. So already we have discussed about that it is the erythroblastosis foetalis. Now, right now. So here in the NEET questions, so what type of questions are asked in the NEET? So right now, when the marriage it is that takes place in between the RH positive female and RH negative male, RH negative male. So what type of, it is a second child, it will be safe or not? So see this one, here it is the 1 it is the, so it will be acceptable or not? It is acceptable or not? So here it is already RH antigen, it is present. So there will not a problem for babies. So because the already Rh antigens are there, so that means this is not a react with any Rh antigens. So that's why there is no production of antibodies. So that's why it is allows the marriage in between this one Rh positive and Rh negative right now. So which type of okay. So which type of marriages should not be advised? So here it is the, you can see this one. Here it is the Rh positive female and Rh negative male. It is the acceptable marriage or not? Right one. Second one. Here it is a female, it is the Rh negative, and men it is the Rh negative. It is acceptable or not? So third one, Rh negative female and Rh positive male. It is acceptable or not? So here it is the female, Rh positive and negative, 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 and negative, positive, and right now here it is the negative will be there and positive will be there, positive, positive, right. Rh female it is a positive, and here it is the Rh positive male, Rh positive male. So here in this one 4 type of marriages. So which marriage it will be compatible, it will be takes place, or incompatibility it will be takes place. Incompatibility means it is not accepted. So compatibility means it will be allows, that means first child, second child, or whatever it may be, they are the safe. So which marriage it will be acceptable? Either first one, either it is the first one, or the second, or the third one, or the fourth one. Which is the acceptable marriages? So which one it is the childs are the safe? So for example, here positive and negative. Already here in this one female organism, there is a positive D antigen, it is present. So here there is no Rh antigen. So already here it is Rh antigen, it is present now. So that means the mother's blood circulation does not sensitize any antigens. So that's why it will be acceptable. Okay, right now. Here the female and male, both are the negative in condition. So that means there is no Rh antigens are the formed in the mother's blood circulation. Because of the male it is does not have any Rh antigens. So that means the female it is does not expose any Rh antigen. So that's why first child, second child, and third child, whatever it may be, the safe. So here it is also acceptable. This is also acceptable. Next one, Rh negative and Rh positive. So here the female organism, when they carry the pregnancy, in such a time the men it is gives rise the antigens to the mother's body. So here the mother it is sensitized with the Rh antigens, and here mother's blood circulation there is antibodies are the formed. So means the second child it will be lead to the death. So that means this is not acceptable. But it is acceptable. In a which time acceptable? When these couples are decided to the only one child. Only one child. Only one child. So they did not want to the second child. They did not want to the second child. So it will be acceptable. But conditions apply. When these couples are wanted to the second child, the second child it will lead to the death. Second child it will be lead to the death. So already we have discussed about that, how the second child it will be lead to the death. Okay, or you can say that. So there is a prevention methods are there. That prevention methods, IgG antibodies and D antibodies are the given to that mother's body. Okay, right now. So here this marriage is not acceptable for the second child. When these couples are wanted to the only one child, it will be acceptable. But they wanted to the second child, it is not acceptable. So before that getting of marriage and after the getting of the first one pregnancy, so there is a RHD antibodies are the given to the mother. That time only the second child it will be safe. Okay, now So that time the second child it will be safe. Which time the second child it will be safe? When the first pregnancy, it is how the 28th weeks of first pregnancy or the 34 weeks of the first pregnancy. First pregnancy, you can given the IgG anti D antibodies. That time the second child it will be safe. Second child it will be safe. Otherwise, no giving of this one antibodies, the second child it will be lead to the death. The second it will be lead to the death. So at the time of the first pregnancy, we can given this one antibodies. The time the second child it will be safe. When these are does not given, the second child it will be lead to the death. So that means these are does not acceptable marriage, or you can say that incompatible marriage for the second child. Only one child it will be acceptable. Right now, here it is the Rh positive and Rh positive. Already the mother it is the Rh antigen. So what type of reactions are that takes place in the mother's body? There is no any reaction it is that takes place against of this one Rh antigen. So that's why it is the acceptable marriage. It is acceptable means compatible marriage in between this one Rh positive female and Rh positive male. So here it is the importance of the erythroblastosis fetalis and Rh blood typing. Okay, so here any doubt just you can ask. Polygenic inheritance it is also there, but it is that takes the time of the 1 hour. Okay, that's why in the next one class we can discuss about that. సో ఏమైనా డౌట్స్ ఉంటే అడగండి. ఇప్పుడు మనకు కింద నంబర్ స్క్రోల్ అవుతూ ఉంటుంది. సో ఏ డౌట్ అని అడగండి, ప్రాబ్లం లేదు. ఐదర్ ఇట్ ఈస్ ద blood groups or Rh antigen typing or the erythroblastosis fetalis. So Rh blood typing and erythroblastosis fetalis both are the very important for NEET also. Okay, the point it will be important in the NEET exam, there is a 80% of human beings are Rh positive. Just you can note that. So 80% of human beings are the Rh positive, means Rh antigen is present in their blood group, means they are the positive blood group persons. 20% persons only they are the divide the Rh antigen. Okay, okay, you can ask anything from this one Rh typing and as well as the erythroblastosis fetalis.
సో ఇప్పుడు స్టూడెంట్స్కి ఎటువంటి డౌట్స్ ఉన్నా
మీరు ఏమైనా అడగవచ్చు. ఇంతకుముందు ఒక అమ్మాయి మనం బ్లడ్ గ్రూప్స్ చెప్పుకున్నప్పుడు ఆ కందుకూరు నుంచి ఒక అమ్మాయి, కల్వకుర్తి నుంచి ఒక అమ్మాయి అడిగింది, చాలా భలే క్వశ్చన్ అడిగింది. సార్ నా బ్లడ్ గ్రూప్, వాళ్ళ మదర్ బ్లడ్ గ్రూప్ ఏ బ్లడ్ గ్రూప్, తర్వాత ఫాదర్ బ్లడ్ గ్రూప్ ఏ బ్లడ్ గ్రూప్, నా బ్లడ్ గ్రూప్ ఏం రావచ్చు సార్ అని చెప్పేసి చాలా బాగా అడిగిందమ్మ. సో అట్లా అడగండి క్వశ్చన్స్ ఏవైనా. కింద నంబర్ స్క్రోల్ అవుతా ఉంది కదా, సో అది చూసి అడగండి, ఆ నంబర్ కాల్ చేయండి. ఓకేనా, మీరు కాల్ చేసేటప్పుడు క్వశ్చన్ అడిగేటప్పుడు ఆ స్క్రీన్ నుంచి పక్కకు జరగండి, స్పీకర్ నుంచి పక్కకు జరగండి అమ్మ. So this one question, it is also very important for the NEET. So this type of questions are there in the NEET examination. So and as well as the 28th week and the 34th week, it is at the time of the first pregnancy. What type of antigens are the given at the first pregnancy? Means it is nothing but known as the IgG anti-D antibodies. After the delivery of the baby, what type of antibodies are the given? There is a anti-D antibodies are the given. It is the difference between that carrying of the pregnancy and the delivery at that after the delivery. At the time of the carrying of pregnancy, there is antibodies are the different. After the delivery, there is a antibodies injections are very different. Okay. So what time the IgG antibodies are the given? Which week it is the given? That is the 28th or the 24th week. ఇలాంటి క్వశ్చన్స్ ఉంటాయి. అండర్స్టాండ్? అర్థమైంది టాపిక్? మొత్తం అర్థమైన క్వశ్చన్స్ రావట్లేదా? లేకపోతే అర్థం కాక క్వశ్చన్స్ రావట్లేదా?
చూడాలి కొద్దిగా ఎందుకంటే వాళ్ళు
Right, okay, all the very best. So next class we can discuss about that next one topic, either it is a polygenic inheritance or other one sex determination. So that sex determination, it is very very important. There is a different type of process are there. There is a female heterogametic and male heterogametic and haplodiploidy condition and a gene balance theory. It is a very 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 important topic in genetics, either IP purpose or either NEET purpose. Every time in a NEET final examination, from the sex determination, the one question it is there in a NEET final examination. So that's why the sex determination, it is a very important for LAQ, or either it is also very important for that NEET examination also. So after that we can discuss about that sex determination. Okay, right. Thank you.
బాబు షార్ట్ జాబ్ నీళ్ళు
Any doubt? Please ask me, Amma.